Hi, I'm Michael Green, and I'd like to welcome you to this week's Semel Grand Rounds. So this week we have Eric Reavis, who's a cognitive neuroscientist at UCLA and has a unique background in both vision science and clinical research. So Eric is remarkable for having a career in which he's built two very substantial bridges, which I'll explain in just a moment. But let me give you a little bit of background on Eric first. So he did his undergraduate at Harvard, where he studied vision science in the psychology department. Afterwards, he received an intramural research training award and went to the National Institute of Mental Health, their intramural program, where he worked with non-human primates, again with vision processing. He got his PhD at Dartmouth, in which his focus was on visual perceptual learning. So it was after he completed his doctorate at Dartmouth that he built the first of his bridges. He decided that as a cognitive neuroscientist, he wanted to apply the knowledge, the skills, the concepts, the methods that he had developed to clinical questions, and specifically to questions in the study of schizophrenia. So he came and joined us as a postdoc after he finished at Dartmouth. We had a long, well-established research program on perception in schizophrenia. It, in fact, was one we were very proud of. That was until Eric arrived and very politely told us all the things we could be doing much better to study perceptual processes in schizophrenia. Some of these resulting studies will be part of his presentation today. During that time, he also received an F-32 individual NRSA fellowship award. He got very involved with programs at the VA Greater Los Angeles, where he has a secondary appointment. That includes involvement in the MIREC, which is our mental illness research center, and Thrive, which is our homelessness research center. He has appointments in both of those centers. It was then more recently that he built his second bridge. He took the information and the knowledge that he had been acquiring in looking at perceptual processes in schizophrenia and psychotic disorders and tried to build a bridge to our understanding of social processing in these clinical disorders. And he did this with a newly received R01 in collaboration with Carolyn Parkinson in the Department of Psychology. So now he's well on his way to looking at these issues as they not only apply to the clinical disorder, but also to social processing within the disorder. So in summary, Eric is a very remarkable early stage investigator and also an impressive bridge builder, and I'm sure you'll enjoy his presentation today. Eric, it's yours now.
Thank you, Michael. That was a very nice introduction. Let me share my slides. All right, well, thank you again for that very kind introduction, and thank you to the organizers of Grand Rounds for the opportunity to speak to you all today. I have no disclosures for this talk, and I actually want to begin the talk at the end and give you a quick preview of the main points that I hope you all take away from this presentation. The first of those is that each of us has a slightly different way of perceiving, processing, and experiencing visual information in the world around us. And we know that on average, visual information processing tends to be altered in schizophrenia, but there are considerable individual differences within schizophrenia, and those are predictive of real-world functional outcomes. And then I'll show you quite a bit of data suggesting that these individual differences in visual information processing in schizophrenia are actually linked to variability in the structure and functioning of the brain. So to get there, this is the plan for the talk. I'm going to start with some background on evidence of altered perception in schizophrenia, and then I'll talk about why we think perception is altered in the disorder, what the neural mechanisms behind this are, and then I'll speak about some ongoing research that we're doing using a newer, more data-driven approach, and finally wrap up with some conclusions and future directions. So I probably don't need to say much about schizophrenia as a disorder for this audience, but just to remind you that it's a common, chronic, and often disabling mental health condition that affects a large number of people, about 1 in 100 worldwide. It's typically diagnosed early in adulthood, and it tends to result in impaired functioning across a number of different domains throughout adulthood, as well as, unfortunately, a number of poor long-term outcomes. There are three major types of symptoms associated with schizophrenia. There are the positive symptoms, which are the ones people tend to think about when they think of the disorder, such as hallucinations and delusions, but also negative symptoms, such as flat affect and lack of motivation, and cognitive impairments, including impairments in things like attention, processing speed, memory, and of course, perception, which I'll focus on today. And I should note that current treatments for the disorder, such as medications, are relatively effective at targeting positive symptoms, but they do very little, if anything, to ameliorate the negative and cognitive symptoms, which can be among the most disabling. Specifically regarding visual information processing, there's a long-standing literature showing a variety of different types of alterations in visual processing, which occur persistently in schizophrenia. That is, they occur outside of discrete psychotic episodes. And there's quite a wide range of different specific effects that have been identified, ranging from things like sensitivity to contrast and spatial frequency differences, various sorts of perceptual integration processes that differ, differences in motion perception, use of perceptual context, perception of social stimuli like faces, even differences in oculomotor functioning, and finally, differences in visual target detection. And for the sake of time today, I'm going to focus primarily on visual target detection as an exemplar of the type of visual processing alterations that are found in schizophrenia. Part of the reason for that is that altered visual target detection is one of the most long-standing, robust, and widely studied examples of altered visual perception in schizophrenia. And there's a very long-standing literature on this going back 120 years now, as far back as Kreplin, who wrote in 1903 about how individuals with schizophrenia demonstrated what he called incomplete perception of briefly presented visual stimuli during laboratory tasks. And over the last 120 years, there's been a great deal of research on this phenomenon, which has really replicated and extended these early observations in a number of different ways. And during that time, the methods have also evolved from these sort of early studies. And a lot of modern studies use techniques like perceptual masking to study target detection ability in schizophrenia. So I'll explain what that is because it'll be important later. So masking just refers to the disruption of perception of a target stimulus by presenting a mask stimulus, that is a noise stimulus, close in time to the target, typically in the same spatial location. There's various different types of masking. Backward masking refers to cases where the mask occurs just after the target, and it's one of the more robust and commonly used types of masking paradigms. The paradigm is quite simple. In a typical masking task, like the one I have depicted here, the subject's goal is just to name what type of object is presented on each trial over many trials, and say whether it's a bell, a clock, a cup, etc. These targets are presented very briefly, typically for only about 8 milliseconds. And then after a variable delay, which is important, there's a high contrast noise stimulus that appears in the same location. And depending on the duration of the delay, the targets are easier or more difficult to identify. And you can see and quantify that by looking at the individual's data. So here's some example data from 2 actual subjects performing exactly the task that I just showed you. And you can see a few things here. So first of all, when there's a very short delay between the target and mask, both subjects here are just guessing. They're at chance performance, which is around 16% when you have 6 options. Conversely, when there's a very long delay between the target and mask, on the right side of the x-axis here, you can see that both participants are near ceiling performance. They're able to identify the targets nearly all of the time correctly. But in between, there's this sort of S-shaped function, which is well described by this fitted psychometric function, the green line here. And what you can see is that although this function has a similar shape across both participants, it's shifted right to left in one subject versus the other. And this is indicative of individual differences in backward masking thresholds. So we can quantify the amount of time that it takes for a person to process and recognize the object information in this sort of masking task by computing a threshold, which by convention is just the point at which a fitted function like this crosses the 50% correct line. So what you can see here is that subject 1 had a higher threshold than subject 2, and that means that subject 1 required a bit more time to correctly identify the targets than subject 2 did. The higher thresholds here are actually indicative of worse performance. And there's been several decades of research now showing that individuals with a schizophrenia diagnosis tend to have higher thresholds than matched healthy controls. And we've replicated that in the study that I'm going to describe momentarily. But it's important to note that there are substantial individual differences across participants within schizophrenia, as well as within healthy controls. And those individual differences turn out to be rather important for a variety of reasons. One of those reasons was demonstrated actually by Michael Green and colleagues about 10 years ago, which is that you can actually link performance on these sort of arcane-seeming laboratory tasks like backward masking and various other types of visual target detection tasks to functional outcomes. That is, how well people are doing out in their everyday lives, whether they're holding down a job, making and maintaining friendships, living independently, those kinds of things. So through a series of associations, it's actually possible to predict to some degree how well people are doing out in the world from how well they do on these sorts of backward masking tasks, which suggests that that this is measuring something that's quite informative and potentially important. But until recently, we haven't really understood why perception is altered in schizophrenia, why on average schizophrenia samples tend to perform a bit worse than control samples on these kinds of tasks, and why there are so many individual differences. In other words, we haven't really understood what the neural mechanisms behind these effects are. And so in the next part of the talk, I'm going to describe some research we've done over the last few years, looking at potential effects of the structure, connectivity, and functioning of the brain, and their potential influences on visual target detection ability in schizophrenia. So the study that I'm going to describe next was a cross-sectional study that had 3 groups of participants. There was a schizophrenia group, a clinical comparison group of individuals with a bipolar disorder diagnosis, and a matched healthy control group, about 50 participants in each. And all of these participants completed, among other measures, a backward masking task, identical to the one I just showed you, an MRI scan, and also an EEG session. And the first thing we wanted to know from these data was whether the structure of the brain might have any relationship to visual target detection ability in schizophrenia. And we had a couple of regions of interest that we were particularly interested in studying in relation to this question. One of those was retinotopic visual cortex, or early visual cortex, which I'm using as a sort of catch-all term to refer to V1, V2, and V3 collectively. As you probably know, these are the earliest levels of the cortical visual processing hierarchy. And so presumably the structure of these regions is quite important for any sort of demanding visual task, like a backward masking task. The other region that we were particularly interested in was this one here, the lateral occipital complex, LOC or LO, which is on the lateral aspect of the occipital lobe. And it is a region which is selective to objects. That is, it contains neurons which are preferentially tuned to certain kinds of objects and respond selectively to certain objects and not others. We were interested in this region partly because the backward masking test that we used required object identification, so presumably this region would be important for that reason. And there was also some early fMRI evidence suggesting that this region might be particularly important for backward masking as well. So we identified these regions in each individual participant's brain using a functional localizer technique and mapped them out on the cortical surface. And then we were interested in whether there were any differences between the schizophrenia group and the other groups in terms of the structure of cortex of either of those regions. So we analyzed this using the high-resolution T1 anatomical images that we collected as part of each scan, and we processed those images using a standard FreeSurfer pipeline, which, among other things, does a very nice job of automatically delineating the boundaries between gray matter and white matter, shown here in red, and the outer surface of the cortex, shown here in blue, quite accurately with sub-millimeter resolution. So once this is mapped out in each individual participant's high-resolution anatomical scan, and once the regions of interest are mapped out on the cortical surface using the functional localizers, it's possible to look at various metrics that characterize the structure of cortex within each region, most notably the surface area and cortical thickness, which is defined simply as the mean distance between these two boundaries within any given region of interest. So we looked at both of these measures. We didn't see any significant group differences in the surface area of the regions, but across both the retinotopic cortex and LOC, there was a slight but significant thinning of cortex in schizophrenia relative to controls. But what we were really interested in was whether there was any sort of relationship to perception and a relationship to visual information processing in schizophrenia. So we also looked at potential associations between these structural measures and performance on the backward masking task that I showed you. And somewhat to our surprise, we found that, in fact, there were rather strong and significant correlations between the two. So I'm now showing you these plots have the masking thresholds on the x-axis. So smaller numbers are indicative of better performance. And on the y-axis, I'm showing you the thickness of cortex in each of these two regions. And what you can see is that in the schizophrenia group, in both retinotopic cortex and LOC, the participants who had thicker cortex in either of these regions performed better on the backward masking task than participants who had thinner cortex in either of these regions. But this relationship was unique to the schizophrenia group. So we didn't see any such relationship in the healthy control group, nor in the bipolar disorder group. So it was only present in the schizophrenia group. And that suggests a couple of things to us. First of all, it suggests that indeed the structure of cortex for these critical early visual areas is in fact related to perceptual performance and visual information processing in schizophrenia. And secondly, the fact that we uniquely see these relationships in the schizophrenia group and not the other two groups suggests to us that these associations might be driven, at least in part, by pathophysiological processes that are specific to the disorder of schizophrenia, which aren't occurring in the other two comparison groups. So next, we wanted to know whether the connectivity of the brain might relate to visual target detection ability in schizophrenia. And as many of you probably know, there's a lot of different ways to assess connectivity using MRI. We looked at several different metrics of this, but for the sake of time today, I'm going to focus on one targeted connectivity analysis that we did, in which we were interested in the white matter structure of a particular tract in the visual system, which we assessed using diffusion-weighted imaging data and a probabilistic tractography technique. The tract we were interested in is this one here, the geniculostriate tract, which is so called because it projects from the lateral geniculate nucleus of the thalamus to the striate cortex, better known as V1. The tract is also known as the optic radiations. And this is, we were interested in this tract because it's the primary input pathway to the cortical visual system. Most of the information coming in from the retina is passed on to cortex via this path from the thalamus. So presumably this pathway is quite important for any sort of demanding visual processing task like backward masking. And we were interested in whether there were any differences in the structure of this path in schizophrenia and whether there were any relationships between the structure of the path and visual information processing in schizophrenia. So we identified the location of this pathway in each individual subject's brain using a probabilistic tractography technique with diffusion-weighted imaging data, in which we used the probabilistic tractography algorithm to identify the most likely path through white matter, connecting the thalamus, shown here in blue, to early visual cortex, shown here in green. And what you can see on the left is an example from one participant showing the path identified by the tractography algorithm, here in red. And on the right, I'm showing you for comparison the known anatomical pathway of the optic radiations from a figure from Gray's Anatomy here. So you can see that there's quite a nice correspondence between the two. And we were able to identify this pathway reliably across participants in this sample and localize it in each individual's brain. Once we did that, we looked at several different measures of the connectivity strength of the pathway, including a couple of diffusion-related measures, fractional anisotropy and mean diffusivity, which each characterize essentially the degree to which water molecules within the pathway, within the tract, are constrained in the directionality of their diffusion by myelin in the axons of the pathway. And also we looked at the volume of the tract, that is just how much space it took up in the brain. We didn't actually find any mean group differences for any of those metrics. So there was no significant group difference between the schizophrenia group or the other groups in any of these measures of connectivity strength. But again, we were largely interested in whether there was any relationship between the connectivity of this pathway and visual information processing. So we also looked at associations between these measures and backward masking performance. And when we did, we saw something very similar to what we saw in the cortical thickness data. Here again, I'm showing you the masking thresholds on the x-axis, where lower numbers are better. And now the volume of the optic radiations on the y-axis. And what you can see is this same sort of relationship that we saw with the cortical thickness, where individuals who had larger optic radiations in the schizophrenia group performed better on the backward masking task than those who had smaller optic radiations. But again, this relationship was unique to the schizophrenia group. So we didn't see this sort of relationship in the bipolar disorder sample, nor the healthy controls, which again suggests that, first of all, the connectivity of this particular tract within the visual system is associated with visual information processing in schizophrenia and backward masking performance specifically. But the fact that we see this uniquely in the schizophrenia group suggests again that there may be a pathophysiological process that's driving this association, at least in part, in the schizophrenia group relative to the other groups. So next we wanted to know whether the functioning of the brain might relate to visual target detection ability in schizophrenia. There's a lot of different ways to look at this, and we looked at a few different metrics, but I'm going to focus on two of them for the sake of time today. The first of those was an assessment of neural tuning precision, which we used a multivariate pattern analysis approach to analyze. So as I mentioned earlier, the lateral occipital complex is defined as a region that contains neurons which are preferentially tuned to particular kinds of objects. That is, they respond selectively to certain kinds of objects and not others. And that's thought to underlie our ability to differentiate and recognize different types of objects. Consequently, the degree to which these neurons are precisely tuned varies across individuals. And presumably that's important for tasks like the backward masking task that I showed you, where people need to detect and recognize specific objects. So we wanted to characterize the precision of neural tuning, which of course we can't do directly using fMRI because we can't measure tuning of individual neurons using fMRI, but we can infer an indirect measure of object tuning precision using patterns of activity evoked during fMRI. So when we show people different pictures of objects, like different chairs and different cups, where it's just a passive viewing paradigm. People are not doing backward masking or any other sort of demanding task. They're just looking at pictures. We can actually look at the degree to which patterns of activity within a region like LOC are more or less differentiated during the perception of different types of objects as an index of neural tuning precision, because each neuron or each voxel within the region contains a bunch of neurons which are preferentially tuned to particular objects. And so if the neurons within a given voxel are more selectively tuned, where say they respond to this chair but not that chair, then we would expect more differentiated patterns of activity than if neurons are more broadly tuned and they respond similarly to different but similar objects. So we can quantify the degree to which the patterns of activity within a region like LOC are differentiated using a multivariate pattern analysis technique. And when we did that, we didn't find group differences in the precision of tuning in LOC, but when we looked at relationships between neural tuning precision and backward masking performance, we found something quite similar to what we saw with the structural data. So now I'm showing you, again, masking thresholds on the x-axis, and now our index of neural tuning precision on the y-axis, where higher numbers are better. And in the schizophrenia group, you can see a similar sort of relationship to what we saw with the structural data, where individuals who had a higher index, that is more an index showing more precise neural tuning in LOC, performed better on the backward masking task than individuals who had an index suggesting less precise neural tuning in LOC. But here we saw this relationship in the schizophrenia group and also in the healthy control group. There was not a significant relationship in the bipolar disorder group, although the trend was in the same direction. So here, this again suggests that there's a relationship between the functional properties of the LOC, that is neural tuning precision, and visual information processing and backward masking performance in schizophrenia. But the fact that we see it now in schizophrenia and in the healthy control group suggests that perhaps the variability that we're seeing here is not driven so much by pathophysiological processes, but by normal variability that occurs throughout the population in this case. Now, I've been talking a lot about MRI up to this point, but I'd like to also make the point that these types of relationships between the brain and perception are not unique to MRI and to the method. So more recently, we've also been looking at an EEG measure of brain activity and its potential relationship to perception in schizophrenia. The particular measure we've been looking at is EEG peak frequency of the alpha band at rest. So when you have people do an EEG recording and have them just sit quietly and do nothing, you tend to see a very prominent oscillation in the 7 to 13 hertz range that looks like this. And is a very strong oscillatory waveform in the EEG signal. In fact, it's so strong that it was the first waveform identified using EEG, and that's why it's called alpha. So it's quite easy to measure. You just have people sit quietly while you record EEG. There are actually individual differences in the frequency of these oscillations. So some individuals have slightly faster oscillations than others. And in recent years, these individual differences in the frequency of resting alpha have actually been linked to a sort of perceptual sampling rate in healthy samples, where in healthy participants, the rate of oscillations in resting alpha is actually correlated with perceptual processing speed. And it's now thought to reflect a sort of refresh rate of perception, the sort of speed of perceptual processing in individuals. There's also been some recent work in schizophrenia by other groups suggesting that on average, peak alpha frequencies tend to be a little bit slower in schizophrenia, and that individual differences in schizophrenia are associated with performance on tasks of cognition and attention. So we were interested in whether individual differences in resting alpha frequency might also be linked to backward masking performance. And we happen to have resting state EEG in these same participants from the study I've been showing you. Lauren Catalano, a recent CDA recipient with our group, has been analyzing these data recently and has found some really interesting results that really parallel the results that we saw in the MRI data. So now I'm showing you, again, the masking thresholds on the x-axis of these plots, and now the peak alpha frequency for each individual in hertz on the y-axis. And what you can see is that there's, again, a significant relationship in the schizophrenia group where individuals who had faster alpha oscillations at rest, when they're not doing any sort of visual task, they're just sitting quietly, those individuals tended to perform better on the backward masking task than individuals who had slower alpha oscillations at rest. But we see the same sort of relationship in the healthy controls as well, although not in the bipolar disorder group. So this, again, is an example of a case where the functioning of the brain is related to visual information processing and performance on this sort of backward masking task in schizophrenia, but also in healthy controls, which again suggests that this may be reflective of sort of normal variability that exists across the population that's driving these correlations. Okay, so just to summarize the first part of the talk, I've now shown you 4 examples of brain-related metrics that are all associated with visual information processing in schizophrenia. Those include structural metrics, connectivity metrics, and 2 kinds of functional metrics. And I think there are a couple of general conclusions to draw from this. One is that it's important to keep in mind that both illness-specific and illness-non-specific factors can contribute to the effects that we see in clinical samples. And the fact that we see some measures that show unique associations in schizophrenia and others that don't is perhaps reflective of the fact that the overall effects are driven by a combination of condition-specific and condition-non-specific factors. Secondly, I think the fact that we see various different measures that are associated with this seemingly simple task is a sort of cautionary note in our in that there can be a lot of different factors that influence even a relatively simple seeming phenomenon. So we tend to want to look for single explanations for a given effect, but in fact, even relatively simple things like backward masking can be influenced by a variety of different neural factors. And so it's something to sort of keep in mind as we look for neural correlates of other phenomena. So going back to the model that I showed you earlier, I've now shown you some evidence that the structure, connectivity, and functioning of the brain can are all related to backward masking performance, which in turn is related to functional outcomes through a series of associations. In the next part of the talk, I'm gonna change gears a little bit and talk about more recent work that we're doing in which we're interested in understanding whether we can go more directly from the functioning of the brain to functional outcomes by developing biomarkers of functioning from fMRI data. So this is a different approach and one that I'll describe next. So this approach differs in the fact that it's more data-driven and naturalistic. I'm going to describe work from an ongoing project in which we're hoping to use methods to link perceptual information processing to social functioning in schizophrenia in the hopes of developing a biomarker. I'll just first mention why we think it's important to identify a biomarker of social outcomes in schizophrenia. Firstly, difficulties in forming and maintaining social relationships are quite common in schizophrenia and contribute to reduced quality of life, and we need better interventions for this. However, the types of outcome measures that we typically use to study social functioning tend to take a long time to change. And that's because when we're asking people about things like the number of friends they have and the amount of time they spend with those friends, you can imagine that that takes months or years to actually change following an intervention. And for most clinical trials, it's not really practical to wait around for a year or two to measure the endpoint and see if an intervention worked. So this makes it difficult to measure the efficacy of potential new interventions. And we think that if it's possible to develop a biomarker of social outcomes that can index these kinds of changes on a more rapid timescale, that could really help to facilitate research and intervention development in this area, because it might be possible to use such a biomarker as a sort of surrogate endpoint in clinical trials in the efforts to develop new evidence-based interventions. Now, you might be wondering what perception has to do with this, because it doesn't necessarily seem like a perception-related question at all. But in fact, there's a growing literature in social neuroscience that suggests that these things might be quite related. And a lot of this work has been done by my colleague, Carolyn Parkinson, who's in the UCLA psychology department. And she and other social neuroscientists have shown quite convincingly in healthy populations that people who have a more similar experience of the world, which you can index using fMRI, are more likely to form social ties. That is, that we tend to affiliate with other people who see the world in a similar way. And I mean that quite literally. So I've just told you over the last half hour about how we know from a century or so of research that people with a schizophrenia diagnosis tend to perceive the world a bit differently than healthy samples on average. And we thought that maybe these things are actually related. And perhaps if on average, individuals with a schizophrenia diagnosis have a slightly more idiosyncratic experience of the world, that might make it harder to find other people who have a similar worldview who it's easy to connect with and form social ties with. So we're now testing this idea in an ongoing study. And we're using methods from social neuroscience in order to index the degree to which different individuals experience the world similarly. And fortunately, Carolyn and other social neuroscientists have developed some very clever methods to do this. And there's a number of different ways to accomplish this, but the most common method, and the one that we're using, is known as intersubject correlations, which are typically measured during a naturalistic perception task. And by naturalistic perception, I mean, essentially we have people watch TV while they're getting an MRI scan, which is about as naturalistic as you can get when people have to lie very still in a small confined space for 40 minutes. Now, of course, we don't just turn people loose with the remote control and have them watch what they want, but we have each participant watch a selected series of video clips, which are usually selected to be somewhat ambiguous and capable of eliciting slightly different reactions in different people. So the way these studies typically work is that each participant comes into the lab and watches this same set of videos in sequence, and they never interact with the other participants at all. They just do the MRI scan and then go home. But what we can do during data analysis is compare the degree of similarity in the processing of these naturalistic stimuli across different pairings of individuals. And the way this is typically done is that you take the data from each individual and you parcellate the brain into a predetermined set of regions using an atlas. So you have the same set of regions in each subject, and then you can calculate the mean activity in each of those regions as people are watching the video clips. And then during data analysis, you can look at the cross correlations of these time series for each region, for each possible pairing of participants, even though they never interact with one another. And from that, you can calculate an R value of the time series correlation, which gives you a similarity metric of the degree to which a particular brain region responded similarly to these video clips in a particular pairing of participants. What the social neuroscience literature has shown in healthy samples is that people tend to have much more similar responses to these kinds of stimuli to people they're closely socially connected to. So if you take the brain response of an individual and you look at the correlation between their brain activity and their friend's brain activity, it tends to be quite strong. If you look at the relationship of their brain activity to the similarity of their brain activity to their friends of friends, it's still correlated, but weaker correlation. And then if you look at people who are not really socially connected, there is actually sometimes an anti-correlation in the degree to which their brains respond in a similar way. So this gives you a sort of metric of the degree to which people see the world in a similar way. And we think that this approach might be helpful in trying to understand and measure social functioning outcomes in schizophrenia. I probably don't have to really say this, but just to reiterate that this is a very different and much more data-driven way of studying relationships between perceptual information processing and functional outcomes in schizophrenia. And we think it potentially has a couple of advantages. One of those is that as a more naturalistic approach, we think it might better capture the sorts of individual differences that people have in the way that they perceive the world, which better correspond to their day-to-day experience. So in our typical laboratory tasks that we have used in the past, like backward masking, we can really only capture one very sort of narrow type of visual processing. But when we have people doing more naturalistic tasks like this, where they're free viewing a sort of complex dynamic stimulus, we think that that's probably going to capture variability across a lot more of those different domains of visual processing that people will use to perceive these stimuli, and in a way that's sort of more similar to the way they perceive the world day to day. Another advantage of this is that it's a lot less demanding and boring for participants than traditional perceptual assessments, where we have people doing many trials of really simple tasks, because people just have to sort of lie still and watch TV for a while, which people tend to like a lot better than doing boring psychophysical tasks for hours on end. So we're currently 2 years into a 5-year research project in which we're using this approach in order to try to understand social functioning in schizophrenia. And we have a few hypotheses for this study. First of all, we hypothesize that on average, intersubject correlations will be lower within schizophrenia than in controls. And that hypothesis is really grounded on the longstanding perception literature that I started the talk describing, which suggests that perceptual experiences tend to be a bit more idiosyncratic in schizophrenia. Secondly, we expect that social functional outcomes will be associated with the normativity of ISC scores in schizophrenia. That is, with the similarity of similarity of each individual's brain activity to the mean of the control group will be associated with social functioning outcomes. And that prediction is based on the social neuroscience literature, which suggests that it's easier to form and maintain social connections with other people who have a similar experience. And if you're closer to the population mean in your personal experience, then that makes it easier to find and maintain connections with other people. And lastly, we think that intersubject correlations might provide a useful biomarker of social functioning in schizophrenia. That is, that we hope to be able to develop a multivariate model to predict individuals' social outcomes from intersubject correlations. So I'm going to show you some preliminary results from this study. Again, we're 2 years into a 5-year study, so all this could change, but so far we're seeing effects that are broadly consistent with the study hypotheses. And the data I'm showing you now have been processed by Lisa Shen, who's a very talented, currently research assistant associated with the study, but soon to be PhD student in the psychology department starting, I think, next week. And what these data show is that, first of all, we do see I'm showing you on the top row here the schizophrenia group and the within group mean intersubject correlations by region. And on the bottom row, the healthy controls. And what this shows is that although we see a similar sort of distribution of significant correlations here, the effects are overall weaker in schizophrenia, which is what we would expect if perceptual experiences are a bit more idiosyncratic across individuals in the schizophrenia group than the healthy control group. So far, this is consistent with the hypotheses, the first hypothesis of the study. We're also starting to look at emerging associations between the normativity of the ISC scores and social functioning outcomes. And so when we look at the schizophrenia group and the normativity of the ISC scores, that is how similar each individual's brain activity is to the mean of the control group, those participants who have ISCs who have brain activity that is more similar to the mean of the controls tend to have higher social functioning outcome scores across all of the regions here that you see in red. And we are seeing a similar pattern of activity of associations when we look at specific measures of social connectedness and also loneliness in this sample. So this so far is consistent with the second prediction of the study. Although I should note that we're still very early in data collection. So of course, we have a lot of data left to collect, and these effects could change over time. So just to wrap up, I hope I've now convinced you that we each have a slightly different way of perceiving, processing, and experiencing visual information, and that we have useful metrics that allow us to quantify this using both traditional measures like backward masking and also newer measures like intersubject correlations. Second, I've shown that on average, visual information processing is altered in schizophrenia, but there are a lot of individual differences across individuals, and those individual differences are predictive of real-world functional outcomes. And lastly, I've shown you data suggesting that these individual differences in visual information processing are linked to variability in the structure and functioning of the brain. So going forward, I think it's going to be important for us to use two sort of complementary approaches to link visual information processing and functional outcomes in schizophrenia. First, there's the traditional approach, the highly controlled approach, which has been very important for the last century and is going to continue to be very important for a variety of reasons. For one thing, it gives us a lot of information about particular mechanisms, and it helps to identify particular intervention targets, both at the neural level, but also at the psychosocial level as well. But there's also this newer emerging pathway, which is using more naturalistic and data-driven approaches to link brain activity and functional outcomes. And I think this is going to be important in that it may produce new and unexpected findings, and also potentially some practical tools like biomarkers. But I really see these two pathways as existing in parallel with one another and informing one another. So I think we're likely to find effects using these newer approaches that we're going to want to go back and investigate further using the traditional method. So I see these two paths as feeding into one another. And a good example of that is from the data I just showed you, where we are seeing effects in the intersubject correlation data that suggests that there's a difference in processing in this sort of lateral pathway of visual processing, which has recently been dubbed the who pathway, in contrast to the what versus where processing streams of the visual system. And the data from the intersubject correlation project suggests that there may be differences in the way that information is being processed in regions of this pathway specifically, like the posterior STS. And I think we're going to want to go further into the mechanisms behind this by looking specifically at functioning within particular pathways like this one. Another important area for future study, I think, which we haven't yet pursued, but which I think would be will be important in the future, is to try to extend this research to the study of other conditions, because we know that there are other conditions that are associated with both altered perception and differences in social functioning. Autism spectrum disorder is the most obvious of these. And I think that similar kinds of methodological approaches might be informative for research on other conditions such as ASD. And so I hope to be able to extend this work in that direction in the future. And as an illustration here, I have an image, a self-portrait by an artist with an ASD diagnosis, who I thought it was very interesting in her artist statement about the creation of this work. She actually talked about how her unique sensory experiences influence her life experience in art. So I'll end there. I just want to quickly acknowledge the NIH for its support of the research that I showed you today, and also the many colleagues who contributed to this work over the years, most notably Michael Green and the Green Lab, Carolyn Parkinson and her lab, and also Lauren Catalano and Lisa Shen, who processed some of the data that I showed you today, as well as the many other people who've worked on this research over the years. So thank you very much, and I'd be happy to take questions.
Eric, that was outstanding. You did a masterful job of illustrating the bridges that you've constructed, going from your grounding in vision science and cognitive neuroscience, applying it to understand perceptual processes in a specific psychiatric disorder, in this case, primarily schizophrenia, and then extending it from sort of perceptual processes to social functioning, the sort of disability aspects of things. So just a huge sweep from neural structure, neural function to perception, all the way to the kinds of elements that come into our assessment of disability. So a really outstanding job. We've already have a couple questions, so let me start going through those. The first question is one about sensory modality, about whether the kind of data that you showed has any correlations with the kinds of individual differences in auditory processing.
Yeah, that's an excellent question. And I obviously have sort of focused on vision here, partly because I'm trained as a vision scientist. So that's been sort of my area of specialization. But obviously there's a very large literature too on auditory processing in schizophrenia, and a lot of paradigms that are widely used, like mismatch negativity, that show differences in auditory processing as well. Unfortunately, we haven't really been able to look at the relationships between the two and whether these are sort of separable differences between the modalities, or if there are sort of common factors that cut across both auditory and visual modalities. But I think that's a really important question for the field to address going forward, because there's likely to be some overlap in both the sort of mechanisms that are contributing to these types of alterations in perception in the visual and auditory domains. And I think that going forward, we're going to want to sort of measure these things together to a greater extent than we have in the past, where we've tended to focus on one versus the other, because I think that will be much more informative. Look at both. In the later data set that I talked about with the intersubject correlations, that is a multimodal paradigm. So we're using video stimuli that have both auditory and visual information together. And so we hope to be able to do a little bit of work to sort of pull apart potential influences of the auditory and visual information. But it's not the best paradigm to do that, because the two are occurring together. So I think we're going to want to also follow up and hopefully look at differences and similarities between visual and auditory processing in schizophrenia using highly controlled methods as well.
Eric, we have a three-part question. Why don't we start with the first part and see where it takes us? I'll read it to you. Did you look at what kind of work school success that normals had with worse ISC scores, which might help those with schizophrenia find work school that is helpful to them and more likely to find success with functional outcomes? Yeah, so that's an interesting question. I think, so we're still quite early in data analysis with that data set.
So we haven't done a whole lot of exploratory analyses of the data yet. We do plan to look at the relationships between the ISC scores and measures of functioning in the control group as well. So we have the same measures across all participants. And I think that'll be a really interesting thing to look at in the future. We just haven't looked at it yet, so I can't really speak to the results yet.
So let me give you the other parts of the question. What were the number of normals versus patients in the task? I think that's in reference to the R01, since the numbers were given for the previous studies, but I don't think it was clear for those, for that part of the presentation. And could MDs predict outcomes for schizophrenia as well as the ISC scores? Oh, that one's a little complicated. I think that means predict outcomes based on other factors, and compare those to the ISC scores.
Yeah, so as I said, the data collection is ongoing for that second study. So the samples that I showed today are quite small. I think we're now up to about 40 participants in each group. We're expecting in the future to have 200 in the schizophrenia group and 100 in the control group. And actually the data analysis that I showed you today was done a few months ago, so it's an even smaller sample there. So I think it was around 25 or so in each group, which is pretty small to really look at these sorts of associations. So I think we're going to need to wait until we have a lot more data to draw any sort of definitive conclusions from the data. But I think it's encouraging that we're seeing effects that are similar to what we predicted. And then, I'm sorry, what was the last part of the question?
I hope I'm interpreting the question correctly. I think it's a question of whether the ISC scores will predict, as well as other clinical factors, functional outcomes. I think that was what was intended, and I hope I got that right.
Okay. Yeah, I mean, I think that's a very interesting question, which I don't know the answer to yet, but I think it's something we'll be able to look at in that data set, because we do have a lot of other measures that we can sort of look at whether the ISC scores are better at predicting outcomes than various other measures were not. So once we have more data, we'll certainly look at that.
And we have a regional specificity question. Were the effects you observed with cortical thickness, parentheses correlation with masking threshold, only observed in LOC and earlier visual regions? Might it be an effect related to global thinning of the cortex in schizophrenia?
Yeah, that's a great question. So we did look primarily at those two regions because we had an a priori interest in those regions. But we also looked at motor cortex as a control region, and we didn't see any associations with the thickness of motor cortex. We didn't end up doing a whole brain analysis because we were trying to limit the number of correlations that we were doing to sort of avoid having too many statistical tests. So, but I think we did, so we did see specificity to these visual areas relative to other regions, specifically motor cortex.
And Eric, I think we have time for one more question. Can you comment on whether these perceptual disturbances are linked to potential outcomes of the disturbances? For example, interpretation biases. So I think this means sort of perceptual interpretation biases. Has that been examined? This would seem to have implications for potential treatment targets.
Yeah, that's an interesting question. I'm not aware of too much research that has actually looked at those sorts of relationships, but I think it's an interesting thing to follow up on. Because, yeah, presumably there are sort of downstream effects of these perceptual alterations of various types, and that could be one specific example of that. But I'm not aware offhand of any studies that have looked specifically at that particular association.
Right. So Eric, with that brings us to the top of the hour, and a really terrific presentation demonstrating this sort of the reach that you've had in answering these very fundamental questions about how perception occurs in clinical disorders and how that implicates different other processes and leads to the steps towards functioning and eventually helps us to explain and hopefully improve disability. So thank you so much. It's been a great pleasure to listen to you as always, Eric.
Thank you. And thank you everyone for attending.